These are the third set of notes on the IB internal assessment for biology. In this section of notes, we'll talk about the conclusion section. Just like the other sections, it will be scored from 0 to 6. For the conclusion section, there are only two aspects that you will be assessed in. The first one is the conclusion justified and relevant to the research question and fully consistent with the analysis presented? So this is where you're going to be interpreting all the math from your data analysis section. You're going to also need to discuss the impact that any equipment uncertainty would have on your raw data and experimental results. The second aspect you'll be scored in is your conclusion and whether or not it's justified through relevant comparison to the accepted scientific context. Essentially, you're going to try and explain your lab scientifically here. It's important that you find credible sources and that you cite your support sources both in text and in the works cited page. In your conclusion section overall, what you're doing is explaining the lab both mathematically and scientifically. So we'll talk about all the things that would be included within those explanations. You do want to try and avoid the word prove. Your data may support a conclusion, but it is unlikely to prove anything. So please don't use the word prove in your lab. When you are interpreting your data mathematically, this is when you're reflecting on all the data tables and graphs that you've displayed in your data analysis section and explaining all of those things. Do you have any patterns or trends that you can see in your data? For example, does the R value show a positive correlation between the variables or a negative correlation between the variables? And how strong is that correlation? So that's where you would talk about R. Are there any results that don't seem to fit the overall pattern or trend? For example, do you have outliers? So you need to try and explain why you know you have outliers, if you have any, and what could be possibly explaining these outliers. Is there a reason for these outliers to have occurred within your data? How much trial variability is there? So this is when you can look at things like the average value and the standard deviation around that value. You can look at your standard deviation error bars on an average. You can discuss the size of the error bar relative to the actual bar itself. This is going to be explaining how reliable your data actually is. The more variable data is, the less reliable it would be. You're also going to be talking about your statistical test here. So when you're looking at your standard deviation error bars, how does it lead you into your stat test? Do the error bars overlap? Do they not overlap? And then explain what those mean. Again, this is going to lead you into being able to discuss the statistical test that you chose. If you used a trend line and standard deviation error bars, then do the error bars touch the trend line? And if they do, what does that mean? If they don't, what does that mean? We don't do this a lot in bio. Most of the time when we put error bars on a graph, it's on a bar graph, but there are circumstances where you would put an error bar on a scatter plot, for example, and then how you view that information would be slightly different. Next, I'm going to click on this link, and it's going to be a link to Bozeman Science, Mr. Anderson, and he will just kind of explain a few mathematical concepts.
Hi, it's Pauline. Hi, it's Paul Anderson, and this is Science and Engineering Practice 4, Analyzing and Interpreting Data. After you've done an investigation, you're going to be presented with a lot of data, and it's important that you learn how to analyze that. In science, data analysis is important because it allows us to derive meaning from the data that we've collected, and in engineering, to test solutions to problems. And so basically, data analysis is important for the following reason. This is Charles David Keeling, and he spent his whole life collecting data on the atmospheric carbon dioxide, how much carbon dioxide there is in the atmosphere. And he did this on the Mauna Loa Observatory in Hawaii. And this is just a portion of the data that he collected as a scientist, just a few years, 58, 59, and 60. And when you look at this, it makes no sense to me and probably makes no sense to you. But if we organize the data correctly and we graph it, you get what's called the Keeling curve. This shows how atmospheric carbon dioxide has increased over time. Again, that's going to account for a lot of the global warming that we're seeing because a lot of that carbon is coming from us. And so it's important that we can put the data together and then present it in a way that we can make meaning from that. And so the goal in science classrooms is to analyze data. We do that by, first of all, organizing and graphing it, and then interpreting the data, making sense of the data, evaluating it, laying it against our theories, laying it against our designs, and then we can use mathematics and statistics to do a lot of that. And so the progression in school of analyzing and interpreting data really should start at the elementary level. And so again, it's like throwing darts, but getting better and better and better through the years. And so we can break this down by school level. In other words, at elementary school, students should be using a notebook, a science notebook, and they should be collecting data, and everything goes into that notebook. And that could be drawings, it can be numbers, it can be words, but they should get into this idea that they're collecting data and they're going to have to do something with that data. As you move through elementary, they should really get to the level of tables and organizing numbers and organizing data in a table. And so let's say you're doing an experiment on fertilizer and how varying the amount of fertilizer affects plant growth. And so they should get in the idea of using a table to organize that. And it's really good practice to always put the thing that you're changing or the independent variable in the first column and then dependent variable in the second. Graphing would be important at the elementary level. I think it's sometimes hard for students to figure out what kind of a graph should I use. And so you could give them quite a bit of scaffolding. You could say, I want you to graph it in a certain way at elementary level. But as they move into middle school, you really want the students to do the graphing on their own. And in my class, I use 4 different types of graphs I expect students to understand. And you should know this, that you're going to collect data first, and then you're going to have to figure out what type of graph to do. And so the data comes first, and so you really have to have an understanding of what kind of data you have before you can graph it. And so the 4 major types of graphs I teach are the line graph. We're looking at change over time, and you're going to connect the data points. We would use a scatter plot if we're looking at correlation of variables, so eruption duration and waiting time at Old Faithful. This would be a scatter plot. We're not going to connect the dots because we're just correlating those 2 variables. We would use a bar graph if we're comparing different groups, and then we'd use a pie graph if we're looking parts of the whole. And so you could present your students with data and first of all have them figure out, what kind of a graph am I going to use? As you move from middle school into high school, you want to make sure they understand the difference between an independent and a dependent variable. Independent variable, remember, is going to be the thing that you change in an experiment or I change in an experiment. Dependent variable is going to be what changes as a result of that. Scatter plots are powerful graphs when we're looking at correlation. So I could graph the data, and then you could get them, show them how to draw a line of fit or a curve of fit. Make sure that you start to teach the difference between causation and correlation. And so when you look at a graph like this, it looks like the fertilizer is causing the plants to grow more. You're not always sure, remember, and this is a good example you could share with your students. Scientists find that there's a definite correlation between the amount of ice cream that people eat and the amount of crime that is done in an area. So if you were to graph it, you would find that ice cream versus crime is going to be a nice kind of a relationship like that. It's correlation, though. It doesn't mean causation. Like, what's really causing this? Well, the warmer temperatures in the in the summer are causing people to eat more ice cream and causing people to commit more crimes in the summer. And so there'd be causation here, correlation here. And so you want to make sure when you're evaluating your data, really be hesitant before you jump into the idea of this is causing that. As you move to high school, you can really take a look at the mathematics and the statistics behind the data. And so a great example would be slope. So once we've looked at this correlation of the data, I could simply calculate the slope of that line of fit. So I could figure out the rise over the run, and I could figure out a y value of 0.4 centimeters per gram. And so slope is incredibly important, especially in a line graph when we're looking at change over time. Another thing that students don't usually understand in my class is what's called the correlation value or the R value. Correlation value, and you can do this in spreadsheets or on a TI calculator, basically an R value is going to tell you how good your data fits to a line if you do a regression. And so if in a linear regression, you can have R values, like an R value of 0 means that there's no correlation between the two data sets. 0.6 means there's some, 1 means it's perfect. And we can also have negative correlation values as well. And so if you're to look up here, what is the correlation value? Maybe 0.7, something like that. And so students can actually start to look at their data and say, hey, how close is it to a line? How good is it? They're starting to then evaluate the data. You can move into the area of statistics then, starting with, let's say this is our data set here. Some simple statistics that I think are pretty powerful are the sample size, mean, median, and range. And so right here, the sample size would be 1, 2, 3, 4, 5, 6, 7. So there are 7 data points. That'd be my sample size. My mean or average is going to be 5. Add them up and divide by the total number. And then my median, to figure out median, would be right here. It's going to be in the middle. And if we were to have an even number, it'd be 5.5. You'll find that students are really good at this. Math teachers do a pretty good job at that. And my range right here would be 11. As you get more and more into statistics, you can move into the idea of normal distributions. I think standard deviation and standard error are incredibly powerful. I'll put links to 2 other videos I made on those. If you can get students to collect data and then look at the data, how good is this data? Then you're really not just displaying the data, you're really starting to evaluate it. And I hope that's helpful.
Oh, oops. Okay. A couple more things you're going to discuss in your conclusions section in regards to the math are the equipment uncertainty and the impact that it would have on your experimental results. Remember, you stated the equipment uncertainty in your data analysis section, in your tables, in your graphs, things like that, but here's where you'll actually discuss it. Not all uncertainty will be impactful. So again, you're going to be discussing, is it impactful, is it not impactful, and how do you know? So for example, if you used a scale which has a numerical uncertainty of plus or minus 0.01 grams, but the numbers that you're measuring on the scale are pretty big, are pretty big, for example, 3.23 grams, then plus or minus 0.01 grams doesn't have a lot of impact, right? So that's a small amount of uncertainty on a number that big. However, if you masked much smaller things with the same scale, then like for example, 0.12 grams, then plus or minus 0.01 grams is more impactful on those small numbers. So you would want to think about, how do I evaluate the level of impact? And then you do need to discuss that in this section. Consider, is there a way for you to mathematically calculate how impactful something is? Okay, so again, you may have to do that in this section. For example, you could set up a proportion, right? How much of uncertainty is this relative to these big numbers versus how much of uncertainty is this in comparison to these smaller numbers? You could calculate a percentage of uncertainty, and then that would help you kind of see the level of impact something has. If that number's high, then it is impactful, and you need to talk about what that means for your lab. If the number's low, it's not impactful, and so that also would need to be discussed. If you are using your eyes to count things like bacterial colonies, then although you won't have numerical uncertainty, it doesn't mean you don't have uncertainty in your final count. So you would need to talk about that. What are some things that occurred in the lab that made it difficult for you to count the bacterial colonies, and why does that lead you to be uncertain in your count? Okay, so again, you do have to still explain uncertainty even when your eyes are the actual equipment. If you did something like counted bacterial colonies, but you had done serial dilutions first to dilute it down, then you would have numerical uncertainty in regards to doing all that math as you dilute the solutions down, because ultimately you're counting a much smaller number of bacteria, and then you're going to multiply it to say how much bacteria you estimate was in your original larger, more concentrated samples. So at that point, there is a little bit of math to do to calculate that uncertainty. So if you end up doing something like that, then that is something that you could ask me or Miss Lockwood to help you kind of figure out how you would calculate that. Then, of course, you're going to explain your statistical test. What did your statistical test tell you mathematically? Did you accept or reject your null hypothesis? And what does this mean for your lab? You also need to explain how you know whether to accept or reject it. So did you compare it to the p-value, and what does that mean? Okay, so you have to discuss your specific statistical test here. Did you use chi-squared? Did you use a two-tailed t-test? Did you use an ANOVA? Did you do some other type of statistical test in order to figure this out? So again, all of that needs to be discussed here. You'll also discuss R-squared value. You probably want to discuss that when you're discussing the R values as well. They kind of go together. Okay, so now that you've explained your data mathematically, you now need to explain your data scientifically. So you need to make a detailed scientific conclusion. It needs to be justified and relevant to your research question, and your data needs to support this conclusion. For example, if you get a p-value that says there's no differences between the data sets, yet you think that there should have been a difference between the data sets, then you cannot say that there is a difference, because mathematically you calculated that there was no difference. So now it's up to you to figure out, how can you explain this, right? You didn't get the result you thought you were going to get. It's time to explain it. Your scientific conclusion must be supported by the data that you actually calculated and presented. You do want to state your results. You want to state any final pieces of data that you use to answer the research question. So if average rates, for example, were used, then you'll need to discuss those things. You do need to make sure you are explaining the science and how you know your hypothesis was supported or rejected. The main concept here is to incorporate the previous sections of your lab report, like the methods, the data, into this new scientific light. It's a really good idea to restate the research question for the moderator in this section. They can't score you down if you don't, but you really should do this if possible. In fact, in the teacher support material, it does say that the students should restate the research question in their conclusion. This kind of like refreshes the moderator's memory on why you're doing this lab, and leads you into being able to explain your results in reference to that question. What you don't want to do is go back and like repeat or relist all the steps of the procedure. That's redundant. So you still do have to be clear and concise and try not to be repetitive. You will want to, again, state your final process data with units. So if you had 3 levels of independent variable and you calculated 3 average rates, then you will state the 3 average rates with units. I would probably put them in numerical order. So for example, from least to greatest or from greatest to least, only because this kind of helps to like establish a trend right away. You can see that trend through those numbers. And then again, you need to explain why the lab gave you the results that it did. Did your data support your conclusion? Is there scientific research that you can compare to? And did you cite? This section in general, the scientific explanation, is where students tend to struggle the most. How do you know what's the right amount of science? You have to find the balance. You can't put too much science, then you're listing stuff that's irrelevant. And you can't put not enough because then your report is incomplete. So again, there is a balance and you're going to have to try and figure out that balance for the IA, as well as stay within the word count. So the hard part is what is meant by the term explain, right? What do we mean by the word explain? Explain is to make an idea or situation or problem clear to someone else by describing it in more detail or revealing relevant facts or ideas. The key words here are detail. You have to have enough detail that the moderator understands the science behind your lab. For example, if you're asked why your Alodia doesn't grow well in the dark versus the light, you need to connect the amount of light received to plant growth. So light is your independent variable, but growth was the dependent variable. Make a clear connection scientifically throughout this process. So you talk about the light-dependent reactions. Electrons become excited in chlorophyll A in the thylakoid membranes, and in the process we make ATP and NADPH. Then those ATPs and NADPHs go into the light-independent reactions, or the Calvin cycle, where they will be used to take carbon dioxide and incorporate it into sugars. Then those sugars will be used by the plant cells in their mitochondria to make ATP. ATP that is needed for plant growth through cell division. So connection to mitosis, right? So again, you have to connect it all the way from the independent variable to the dependent variable. And you want to do this for all the levels of independent variable. Light would be expected to increase all of these reactions, whereas darkness would be expected to decrease all of these reactions. It is your job to explain this clearly and thoroughly to the moderator. And again, be sure to provide references. That's it for the conclusion notes.